Welcome to this class on Neuroscience of Human Movement. This is part 10 of the discussion on primary motor cortex. So, we have been discussing primary motor cortex and its contribution to motor function and dysfunction in when there is a lesion in the primary motor cortex. So, in the last class we discussed what could be the role of lesions of primary motor cortex on movements. So, let us review that concept. So, in today's class we will be reviewing lesions, we will discuss the differences in recovery timing between animals and humans across species, and then we will discuss the differences between the two types of lesions, those that happen in the primary motor cortex or in the upper motor neurons. So, let us remember the discussion from the previous class. What are the various factors that affect recovery after lesions? What are the various things that affect how devastating is a lesion? What are the various things? We said this location of the lesion is obviously an important factor, then volume or size of the lesion is another important factor. Lesions of comparable or normalized size produce more devastating effects as you go up in the hierarchy from small mammals to larger mammals to prosimians to macaques to great apes to humans and so on and so forth, and the level of skill also affects the extent of dysfunction and the possibility of recovery. So, understanding these things together is a great challenge, and only a deep understanding of this can lead to novel, useful, innovative rehabilitation approaches in humans and maybe development of devices that could be practically useful to these individuals. So, it is not a theoretical, so there is these topics are still a matter of active debate and active research. So, still lots of research continues to be done. We will discuss some of this in the future slides. So, then we said pyramidal syndrome. So, pyramidal syndrome basically is this, it is a misnomer that refers to a whole bunch of symptoms that are caused not just due to pyramidal neurons, but also due to other upper neurons. This usually causes. So, what are the classical signs? The classical signs of pyramidal syndrome, the classical signs of the upper motor neuronal syndromes. What are these? First, Babinski sign. When the fanning of the toes and extension of the great toe happens in an adult, that is called as a positive Babinski sign, which is abnormal in an adult. Then spasticity. What is this? We said what this is. This is rate dependent rigidity. A given load is applied slowly, the resistance is low, and the same load is applied fast, then the resistance is relatively high. This is called as rate dependent rigidity or velocity dependent rigidity that is felt, right. This is called spasticity. And the third classical important sign is the loss of fine movements. Then other things such as contralateral paralysis, then hypertonia and increased tone, that is usually preceded by hypotonia or so what happens as soon as there is a lesion, there is an initial period during which there is spinal shock, there is decreased tone or hypotonia first, then this is followed by a period of abnormal or high tone called as hypertonia. Hypertonia comes but after an initial period of spinal shock, and then increased deep reflexes called hyperreflexia, and practically disappearance of the superficial reflexes. This is what we have seen in the previous class. So, then what happens across species? Across species we said that the effects are more devastating. We said this earlier. What does that mean? So, for example, when there is a lesion in a cat, there are cases when there is practically no paralysis. So, the cat can actually walk quite normally within a few hours after the experimentally induced lesion. It can walk on flat surfaces but not on challenging surfaces. So, how are even these lesions caused in these animals? So, there are a few methods. I will just mention them, those who are interested can check this. One is the suture based occlusion of the middle cerebral artery, the classical approach. So, middle cerebral artery is occluded, causing an infarct, causing a lesion. Another approach is photothrombosis. What this method involves is photosensitive dye such as rose bengal is injected intravenously into this animal, and a specific area of the animal's head is irradiated. How to choose which area has to be irradiated? The irradiated area will lose the cells, right? How to choose which area? The regular stereotactic methods, the regular mapping methods that are used in neurosurgery are used. So, stereotactic approaches are used to choose the area that is going to be exposed to irradiation, causing death of cells. So, using this approach, focal lesions can be delivered even in small mammals such as rodents. And more popular is the approach to use vasoconstrictors. This also has the ability to deliver focal lesions. So, different approaches have different advantages, disadvantages, and comparable how comparable these are to real strokes in humans, etc., are a matter of debate, and anyone who is interested can check this out. Importantly, the following is a question: is there a critical period for recovery? In other words, is there a particular period during which if I train an animal after a lesion, there is a higher potential for recovery? This is crucial because suppose a person has stroke, if you miss the crucial window of recovery, then you are not going to get back that time, and it is not clear what this crucial period is. First of all, is there a crucial period for recovery? The answer is yes. From animal studies, it is now almost accepted that there is a crucial period during which motor activities must be trained soon after a lesion. There is a particular period after which the motor training must be given, and within a particular window there is a greatest effect of recovery. Now, this raises a lot of questions. Does that mean this critical period is the same for all individuals? Is it the same for all strokes? Lots of questions. So, these matters remain a matter of active debate and active research. At least now it is accepted that there is a critical period within which the training for motor recovery or motor activity training must be imparted to patients, so they have the highest probability of recovering their motor function. Now, as to which patient has to get how much, what is the lesion that must get how much, we should decide. So, studies must continue in this area, so we have more clarity. Then that takes us to the topic of what constitutes what causes how much amount of disability, and what constitutes disability and how to recover them. Fundamentally, to remind ourselves of the following, there are two types of motor neurons. There are the upper motor neurons, these are the motor neurons or the corticomotor neurons. These are the motor neurons that make monosynaptic projections with alpha motor neurons in the spinal cord, right. So, for example, if you see this motor neuron, its cell body is here, its axon is travelling a relatively long distance until the spinal segment and is directly innervating a neuron here. So, this neuron is the alpha motor neuron, is it not? So, this alpha motor neuron controls functions in the distal most segments of the upper extremity. For example, the example given here is a person is trying to find out whether the shower is too cold or too hot or whatever, what the temperature is. So, here is the red indicating hot shower, that is a cool tap you are trying to open, and the person is keeping the hand. So, obviously there is, so what happens is that there are sensory endings in the skin that is going to detect the temperature or the thermoreceptors that are going to be detecting, and that is going to end up not necessarily through a monosynaptic pathway, but rather through a polysynaptic pathway. There are several interneurons that take it to the primary somatosensory cortex. So, there are basically it goes through, this is the sensory axon, and then it enters the spinal cord and synapses with neurons of the brain, and then there are several interneurons, and then it continues projecting to the thalamus, and from thalamus on to the cortex, the sensory motor cortex, right, then where it is consciously perceived, and some sort of processing happens here. From there appropriate commands to the motor area is given, and then the motor neuron directly controls movements of the hand. Well, if it is too hot, it is important that you pull out the hand for your own safety, is it not? So that command is going to be given through a monosynaptic projection from the corticomotor neuron to the alpha motor neurons. So this is the pathway. We have discussed these things earlier, and what is widely believed is important. I put this in quote because it is a famous phrase. It is now somewhat well accepted that the alpha motor neuron muscle projection is the final common pathway that controls movements. Let us remember that the alpha motor neuron controls which muscle fibers are going to be recruited, how much force is going to be produced, or in other words the nuts and bolts, the fine details or the fine mechanical details of who is going to participate or which muscle fibers or which motor units are going to be participating in the particular action are decided not by the brain but by the spinal cord. So the alpha motor neuron muscle pathway is considered as the final common pathway, famous phrase from a famous scientist. Check who it said that. So then this alpha motor neuron in the spinal cord, which is called as the lower motor neuron, projects to the muscle and causes particular action. Now importantly, it's one has to be cautious about these interpretations of upper and lower. When you say upper, what do you mean? When you say lower, what do you mean? Well, let us discuss what are the differences between lesions of the upper motor neuron and lesions of the lower motor neuron. What could have, when could lower motor neuron lesion happen? When the alpha motor neuronal integrity in the spinal cord is compromised, you have what is called as a lower motor neuron lesion. So fundamentally different from the lesions of the cortex or the brain, the motor neurons in the cortex or the corticomotor neurons are the upper motor neurons, right. So the upper motor neuron lesions generally refers to lesions of the motor neurons that are above the level of the brainstem or above that hierarchy, right. So let us discuss some of the characteristics. What happens to muscle tone? First, we said this earlier, in the case of upper motor neuronal lesions, initially there is a period of hypotonia or spinal shock that is followed by a period of hypertonia or a period of increased tone. Why is there increased tone? That is a question. Why? Because the inputs that are received by alpha motor neurons are complicated. Not all the alpha motor neurons receive monosynaptic projections. They usually receive, most of the alpha motor neurons in the spinal cord receive inputs from interneurons. It ultimately results in an increase of the tone. It would appear that much of the descending inputs are inhibitory, but in general much of the descending inputs is gone. Removal of these inputs causes a result that the tone will ultimately increase. So it is the loss of the descending inputs or the complicated nature of this descending inputs that cause this increased tone in this. If the alpha motor neuron is lesioned, then basically the muscle does not get any more inputs, so there is no more active contractions of the muscle, leading to decreased tone or reduced tone or hypotonia. And what happens with weakness? Well, if the lower motor neuron, if the alpha motor neuron in the spinal cord is lesioned, there is profound weakness. The effect of weakness is huge, and the paralysis that is caused is flaccid paralysis. Whereas in the case of upper motor neuronal lesions, what happens is that the response is graded. Either there can be paresis or weakness, or there can be plegia or paralysis. In general, this paralysis is, when compared with the lower motor neuronal lesions, is spastic. What is the difference between flaccid paralysis and spastic paralysis? Flaccid paralysis involves weakness, whereas spastic paralysis involves spasticity or a regime of increased tone, velocity dependent rigidity. Usually this is preceded by spinal shock. This we have already discussed. What about muscle size itself? Is there an atrophy? Since alpha motor neuronal projections to the muscles are lost, there is a denervation. This denervation causes a reduction, a substantial reduction in muscle mass, causing what is called as a severe atrophy. So trophic factors are disappearing from the spinal cord, causing atrophy of the muscle. Whereas in the case of the upper motor neuronal lesions, there is no atrophy, because alpha motor neuronal projections to the muscles are already present, and actually they are sometimes more active than rest, right. So because of this reason, the atrophy is usually minimal, and any atrophy that is happening is purely due to disuse. So fundamental difference. In the case of lower motor neuronal lesions, the atrophy is due to denervation. In the case of upper motor neuronal lesions, this is due to disuse. There is no use. If you are not using the muscle, then you are going to lose it. So this atrophy is present also in patients. For example, even if the person has not had a motor neuronal lesion and has been bedridden for some other disease, for example, has not been using the body for using the muscles for a long time, then what happens is that there is a disuse related atrophy. Similar thing is what happens in this case of upper motor neuronal lesion also. So the atrophy is minimal. And in the case of lower motor neuronal lesions, because it is possible that alpha motor neurons could be partially damaged, because of that reason there can be spontaneous bursts of activity from alpha motor neuronal pool. Because of this reason, small twitches of the entire muscle may be felt. These small twitches of the entire muscle are called as fasciculations. These are sometimes prevalent. If the alpha motor neurons are damaged completely, but some branches of the axons are present, then these branches of axons sometimes cause small twitches of individual muscle fibers. So if this happens for an entire muscle, that is called as fasciculation. If it happens for individual muscle fibers, that is called as fibrillations. So fasciculation is something that is felt superficially. Fibrillation happens within the muscle, but not necessarily felt superficially. These are present in lower motor neuronal lesions. These are not seen in upper motor neuronal lesions. And what about reflexes? When you talk about reflexes, when you are stretching a muscle, there is going to be a stretch reflex that is present usually in the healthy intact system. But when there is no alpha motor neuron that could activate this muscle, there is either weak or absent stretch reflexes. So the monosynaptic reflex, for example, the tonic stretch reflex, for example, is going to practically disappear. But in the case of upper motor neuronal lesions, we said what happens, there is hyperreflexia. The reflex amplitude goes up, there is brisk, very quick response, hyperactive deep reflexes, and this is sometimes also accompanied by what is called as clonus. What is clonus? Clonus refers to the situation when the muscle is suddenly stretched and kept at a different length. Suddenly, when this is done, oscillatory reflex responses are produced. Oscillations are produced as response to a sudden reflex causing stimulus. When that is done, sometimes there is oscillatory response, usually at about 7 hertz approximately, give or take. Please check this. So this is called as clonus. This is usually present in the upper motor neuronal lesions, but not in the lower motor neuronal lesions. In the, what about Babinski sign? We said what is Babinski sign? The fanning of toes accompanied by the extension of the great toe is present in upper motor neuronal lesions, but not present in lower motor neuronal lesions. Extensor response is present in upper motor neuronal lesions, but not present in the lower motor neuronal lesions. Then also note that when a particular area of the motor cortex is affected, it is not individual muscles that are getting affected, but rather coordinated movements that are performed by multiple muscles are affected. It is movements that are getting affected or multiple muscles that are affected, resulting in the loss of fine movements in upper motor neuronal lesions, whereas lower motor neuronal lesions, if they are focal, may affect only one muscle or its agonist. So, individual muscle or group of muscles may be affected, because why is this? Because neurons that are performing similar functions are located next to each other. We call this as a muscle field in one of the previous classes, is it not? If that area is compromised, it is possible only one muscle is affected or its agonist or that group of muscles are affected without affecting the other muscles. Whereas in upper motor neuronal lesions, it is the movement or the coordinated activity of all these muscles that may be affected. Some examples, hemorrhage, spinal cord transection, all these things cause upper motor neuronal lesions. And what about lower motor neuronal lesions? This can be due to spinal root lesions or polio, for example. Poliomyelitis is a disease that has been almost eradicated from the world, except for maybe in Pakistan. Everywhere else has been eradicated. Now that disease is a disease of the lower motor neuron, right? So, and there are other examples. So with this, we have discussed the differences between upper motor neuronal lesions and lower motor neuronal lesions, fundamental differences in clinical signs, and fundamental differences in mechanisms of dysfunction. So what have we seen? We have reviewed lesions in today's class, and we have discussed timing in different animals. We have asked the question, is there a critical period of motor training for rehabilitation? The answer is yes, there is. And how to identify that? That's the $1 question, that's the $1 question that we need to answer. And we have compared how or what are the effects of upper motor neuronal lesions and lower motor neuronal lesions. So if there is a critical period for motor training, this gives us an idea that maybe motor training can help improve movements in future. That means it is possible for the brain to restructure itself or somehow realign to perform a new function. That is a revolutionary idea that we will discuss in future class, or we will discuss the role of plasticity in motor recovery in the future class. So thank you very much for your attention.